Yeah, hello, I'm Dominik. I'm from Ibrahim Lab from University of Virginia. I'm also deciphering the mouse intestine, in which I will be talking about current optimization of intravital microscopy of mice intestine with the help of very nice confocal spinning disk system. And when I started this, I didn't have anything apart from this. With a lot of patience, 3D printers, hot glue gun, and access to Amazon, I used a couple of concepts. First one was something like this, where I printed the stage and additional holder for the tissue to stabilize in all axes. Then I went to more sophisticated structures which are isolating the intestine from the body, so there is no any movement from breathing or heartbeat. And so what are the results? One of the coolest videos that I managed to record is wave beta neutrophil inside blood vessel of intestine, which looks like it's walking. So this is definitely one of the coolest things. But apart from it, when I was playing with GFP tagged mice, GFP was tagged to some junctional protein. I noticed with lower magnification that I look at not only crypts, but also slices of villi in living tissue, which is extremely cool because whenever you think about structure of villi and crypts,
30 seconds.
So looking differently, you can look at 3D architecture of how crypts and villi. And I've calculated plenty of symmetry and stuff. And the last thing is going 3D in time. So this is an example of first of very few 4D recordings of the tips of villi in mice intestine.
5 seconds.
Visualized in Siglass, which is very cool software. And I actually brought VR headset with me with posters. So yeah, come and see that.
Dominik has poster number 2. So he'll be out there, and so he will be at his poster tomorrow.
Yeah. This one is gonna have a timer. I'll tell them 30 seconds. It's fair. Okay. Ready?
Hello everyone, my name is Leor. I'm a PhD student at Castillia's lab of computational cell dynamics, and today I'm going to talk about multimodal bioimaging and computational profiling of triple-negative breast cancer. So our amazing collaborator, Maddie Parsons from KCL,
Makes sense.
Has created an amazing data set of a multimodal niche data set. So what we do, we take a tissue sample, we do consecutive tissue sections as we can see above, and each tissue section is then imaged using a different bioimaging technique. So we have H&E, spatial transcriptomics, spatial proteomics, metabolomics, and confocal fraction. And we can see them here below. So this is the H&E, the genes, proteins, metals, and the DNA. And because the consecutive tissue sections are just 4 microns, we can assume that they share the same information in their regions, and therefore we can look at them as the same. So my research focuses on taking this unique data set and then integrating it into a multimodal data set, and then looking at if we can build models to predict response to triple-negative breast cancer and look at the interplay between the different modalities. So for example, if we find that a patient is a non-responder, what is the proteins, and then go to the metals and the genes and see if we find some relationships. And of course, everything connects to the final insight.
30 seconds.
So I'm poster free, and if anything about multimodal data sets, integration, registrations, and analysis interest you, come talk to me.
Great.
Conrad?
Hi, I'm Conrad from the research campus. This is a kind of a web tool I built quickly to make a data set browsable on the web. Kind of inspired by the IDR, and we wanted to have users be able to find their images and just view them in Neuromancer. So I just wanted to make something very simple and see if I can make it also usable at the same time, so you could put in different data sets. So before I knew about BioFile Finder, which I think is also a great tool that does probably all this much better. But the way this works is you have a metadata spreadsheet. This is the typical way to get our metadata from the scientists. There's a link to the czar in there and whatever metadata they want. And then I built this little executable on the command line. You can load in that basically you define a YAML file that points to your spreadsheet, and that points to data on S3. And then you can just basically customize the website, find what columns you have, and how you want them displayed. And that generates this website. The main point here was being able to search by any of the metadata. You can have search box or drop downs to sort of filter on those values. And then if you mouse over one of the images, you get this inspired menu of options, and one of Hernandez Neuromancer. One thing I'll say is Neuromancer, it doesn't quite support multi-channel data out of the box. You have to generate a configuration program, and Parking does that. You have good access to it. So yeah, I'm close. Cool.
Got Erin, Jen, and Tony not next in that order.
To be more efficient, you might want to. It's good at pretending anyway. There you go. All right, hi everybody. Poster I brought is on the cell painting gallery. I'm Erin in the Simony lab and the imaging platform at Broad. So morphological profiling is an imaging technique at scale. The cell painting assay is the most common morphological profiling assay. And happy to go on for a very long time about this, but in the interest of time, I'll say the cell painting gallery is a public data repository that we built and maintain. It has 22 complete cell painting data sets. So this includes raw images, generated intermediates, and all of the analysis files, all the way through profiles, which are vectors describing perturbations. So that the complete data package end to end. So we right now 16 in progress data sets. So lots of active work generating new data. It's currently 770 terabytes of data. It is sort of unique, I think, compared to some of the other large data sets and repositories folks are talking about, and that this is very tightly constrained to a single assay or very close derivatives. It's entirely hosted in AWS, and this is thanks to the fact that AWS pays for the hosting through their registry of open data. So we don't actually have any specific funding for this repository. The majority of the data has come from a single source, generated at the Broad, but we are working on actively bringing in data from other groups. It turns out it takes a heck of a lot more of my time to onboard external users than it does to just upload the stuff where I have touched the data from end to end. So that's a bit of a challenge. And again, like my time isn't actually funded to get other people's data into the gallery. So happy to have discussions about funding data repositories and me. And again, like the majority of this is a single cure, like I've touched almost every single piece of data. I'm working really hard to make it as fair as possible, but it is mostly me spending the time on it. So I have lots of efforts, and I would love to talk to people about my efforts to make it fair and how maybe people could help me make it even fairer. So that's what I've got. Good.
Hi everyone, my name is Genevieve. I'm a master's student in Claire Brown's lab at McGill University, and I'm going to be talking to you a little bit about super resolution algorithms. So what super resolution algorithms do is they take a diffraction limited microscope image and they output a synthetic super resolution image. In my project, I'm going to be using confocal as my diffraction limited example instead as my super resolution ground truth. And really the purpose of this technology is to make super resolution more accessible. It helps overcome the trade-off between having a diffraction limit and having a really complex and expensive microscope. And I just want to emphasize that the point of this technology is not to replace super resolution, but more to supplement it and make it more accessible. So these algorithms exist and have been developed, but they haven't really been compared or benchmarked. So that's what my project is trying to do. I'm going to be comparing 3 deep learning based algorithms and 1 mathematical one, just to see which ones are the best basically, and which one or what the advantages of each are. So I'm going to be using confocal instead image pairs of actin filaments, 2 examples of which are here. And I really want to see which ones perform the best in generating a synthetic super resolution image. And I also want to evaluate across different levels of image quality, such as different pixel size and different in the picture. And then beyond this project, I'm interested in seeing what biological applications can be used with this technology and how can this be deployed in a user-friendly setting. So if anyone has any input on those future directions, I would love to hear. I'm looking for ways to come with that. No.
It's not presentation.
Oh, loaded.
Yeah, joke,
450, it's not my.
Yeah, so.
That's a new record.
maybe.
Yeah, I think we're gonna go without the slides, so.
Or skip to the next and you can upload something smaller.
Yeah.
Maybe
Yeah.
download anyway. Yeah, we can go with the next and then we can wait.
Downloading.
I was thinking about it, right?
Talk about big data.
It is, okay.
That's.
This is not included in my time, right?
So.
We should just jump it.
Yeah.
No, it's not.
Okay.
How many jokes can we get done before it downloads? A sharded presentation.
After Tony, it will be Hiroya, Mark, and Abby.
All right,
You 3, not yet,
Ready?
2.
go.
My name's Paul. I'm a PhD candidate at the University of British Columbia. I'm from Dr. Timothy Murphy's lab. We do a lot of hardware development and software development. So like Dominik, he mentioned that a lot of our things are from Amazon and about 60% for Amazon, 40% from AliExpress, if you know what it is.
I guess with the trade war, we're gonna go about 100% AliExpress in the next few months. So one of the tools we developed is based off of Raspberry Pi. So we use these kind of affordable cameras as single board computer solutions to take scientific images. These are very cheap and very small. We can fit them into many facilities and collect data at scale. So recently there has been development in camera drivers. One is called Lib Camera. So it basically allows you to get raw data from any commercial camera that is adaptable to a Raspberry Pi. Otherwise, you would have to get a commercial camera, which is like quite a few $100. These are most expensive in a lot of microscopes. Normal commercial cameras before kind of Lib Camera is that they have a proprietary image signal processor, which takes data from the sensor, turns it into something that you think it's beautiful. Normally, you can't bypass that. That's directly into the camera. Here for my research, we can do a lot of wide field imaging. So we do intact skull imaging. So we have these genetic animal lines which express calcium indicators, and we can just image directly from the skull. So we use a tandem lens setup to do this wide field imaging. So this whole kind of setup just costs less than $500 overall. And we do a lot of behavior imaging as well. So we take these cameras, we chain them up together to do this 3D behavior imaging and wide field imaging at the same time. Here is a mouse kind of doing a water reaching task. We can see we can capture all of its 3D movements. And since these are very cheap to build, in our facility, we have like 6 of them. So we can see images of that. So we collect about 30 mouse a day, basically, of imaging data. And I'll leave off at this slide here, which is basically all the data I've collected, samples of data I collected from single cell to wide field imaging. Thanks.
US dollar or Canadian dollar? Indian dollar, Indian dollar.
Canadian rupees.
The same now.
Oops. Something. Oh, something. No problem. So hi, my name is Hiroya Itoga. I'm from Nikken, Japan. Every year, everywhere, my boss Shuichi already introduced what is SSBD. So I only talk the topics for the poster. Very important information. The SSBD image color is orange. So please come to discuss with orange guy. And here's the picture of the founding guide. Shuichi already announced the founding guide, but I put the flyer in the registration desk. Please please pick the one founding guide flyer. And next is the scalability. We want to grow up the number of the submissions for the scalability. So we are developing an easy-to-use submission system in the SSBD repository. It's very easy for the essential metadata. Please come to SSBD KNJP submit. You can use the protocol for the submission system. It's already used, but we want to improve the system for the submission system for the next scalability system. And finally is the Globius 2025 in Kobe. I'm one of the local organizer, and I also put the Globius 20 the registration disk. Please pick one flyers because I don't want to bring back the flyers. Okay. Pick all the flyers. My poster number is 11. It's my orange guy.
Oga-san also promises a beer tour in Kobe.
Yeah, that's how you. Thank you, folks.
Identical slides.
Yeah.
Ridiculous.
The median
So-
number of times per OME meeting this one.
So we've seen this cartoon developed plenty of times and we always laugh at it and maybe cry a little bit inside about what this means for us. But I decided to try to think about it, see if I could try to maybe get around this problem. And so the word I want to focus on here on this slide is competing. Maybe these things don't actually compete. Okay. So it turns out if I take a look at TIM, H5, and star shards, actually they fit together pretty nicely. People have done work to cloud optimize TIM files, compile the TIM file metadata at the beginning. You can even do 3D chunks inside a TIM file these days, use GeoTIM. With HF5, you have a user block where you can stuff that TIM metadata, but then you can have the HF5 metadata all consolidated near the beginning. Shard metadata can either go at the beginning or the end, so let's put it at the end of the way of the TIM. And actually you combine that all together so you have a single file. That's not a valid TIM file, a valid HF5 file, and a valid star shard. So in theory, that sounds great, but how do we do it? Well,
It's now kicked me off 3 or 4 times. That's awesome. There you go.
Okay, so good afternoon, everyone. I'm Marco Antonio Hernandez Valenzuela, a PhD student in the National Polytechnic Institute of Mexico. And today I will talk a little about my research that is T cells in the tumor microenvironment of pediatric Hodgkin lymphoma. Lymphoma is a type of cancer that affects B cells, T cells, and natural killer cells. In patients under 19 years old, is one of the most frequent cancers, ranking not Hodgkin lymphoma the 3rd place and Hodgkin lymphoma the 5th place. Classical Hodgkin lymphoma have 4 different subtypes, and each subtype have the tumor cells. That is the Hodgkin red stem cell. That is a cell derived of B cells, but have a different phenotype. And what makes tumor microenvironment in Hodgkin lymphoma unique is that most of the cancers have most of the tumor mass composed by tumor cells. But in Hodgkin lymphoma, only 1 to 10% of the tumor mass are tumor cells. So that's why it's important to study the other 90% of the tumor mass that are immune cells in order to predict the prognosis of the patient or the treatment response. And how what my research methodology is? Well, right now I have 13 samples of Hodgkin lymphoma, and I did immunofluorescence to detect T cells and analyze the images by cell pulse plus Fiji and do that analysis. And the results looks like that. So we first explored the incidence of CD4 and CD8 cells in the tumor microenvironment, and we sort by when we compare the different subtypes that we have, we found no difference between them. So okay, so well. If you want to know more, thank you for your time. If you want to know more, phone me and my poster.
Thank you. Well, my name is Tom. I'm a fan of minimalist slides, apparently, not as much prepared as the others. I wanted to take this opportunity to talk about this thing we're doing. We're starting a new project, often in it, creating a new project, naming it, finding. But really, I would like to emphasize contributing to existing projects that we have in the community is also as important and probably much gives much more value to what we're doing. So I really do have a preference between the two. I'm highlighting one in red. And for that, I unearthed some of my old projects, some cursed projects of merging 3D meshes to solve some problems. If you're curious of why someone would do that, come to my poster. And I really like to compare to newer things I was been doing, contributing to existing projects. And one where a project was loading slices in Blender, and I added a few lines of code and were able to load orthogonal slice and then create animations in Blender. And so really with a tiny bit of work, you can make great things with existing packages versus months of development it would take you for projects that maybe don't always make sense. Yeah, so if you're interested in discussing this topic with me, that's number 16, not on my slide, but well, actually you can find it around. Thank you.
Hello, everyone. My name is Dominik Rodzen-Staff. I'm the lab at McGill University. I'm working under this review.
You don't need to.
You don't need to get it.
Yeah, so I do want to credit my colleague F. Turur. He's done a lot of the work. He wanted to come, he was not able to make it, so I do want to give him credit because he's contributed as much as I have to this project as well. I only have about a minute, so I think the main things I want to go over is the issues that we were told from the Strambio lab. Professor VBL connected me to Caterina, and essentially what she told us was that the main issue was about the metadata in a shared framework. Currently, there are different tools used. Essentially, yeah, here. Currently, there's different tools used, and there's some data loss when converting from proprietary data to community metadata. And essentially what we ended up doing was schemas for LinkML. So essentially we have a proposal for LinkML. It's a YAML-based file that LinkML runs on, and these can create row-grade files and as well as JSON files, ER diagrams, and then we can also create ER diagrams to get to LinkML. So you can get an ER diagram, for example, to LinkML and make a JSON file out of that or anything of that sort. And there's a few other files that are supported with that as well. And essentially what we want to do is carry that out at the Strambio lab through proof of concept. So yeah, our goal is essentially to represent the NBOQ model, which is an extension of the OME model that's used in new applications essentially. So far, we have done the representation of the OME model, which covers about 80% of the applications, and the model covers more of the new applications. So right now, this is essentially the standard. It's all manual workflow, and what we're proposing is essentially a automatic workflow using LinkML, and this kind of represents all that. So I only have about a minute, so if you want to come talk to me, any criticism, feedback, questions would be appreciated. Thank you.
Caterina, Graham, and then William.
Oh, no, we didn't send. No. Thank you. Take care. No. All right, so this is a pitch to use REMBI as a way of creating a bottom-up standard for image data. So REMBI, and the idea is that REMBI can also be used as a way of gathering the community around what are the different aspects of metadata that can be that we need to collect. So different communities can take part, that can actually develop different parts of this, and then we can exchange what other people are doing. As far as this, the part of the REMBI, which is before acquisition, the idea is that there are these modules can be used as the REMBI can be used as a way of creating modular key value pair sets that can be combined together depending on the type of experiment that people do. And then we can exchange this and see if any kind of standard emerges. So the idea is that each one of these pieces can be a piece of the puzzle, and then depending on different type of experiments, we can have exchange different type of models. With the we have a small project that was proposed last year at OME. Now Netherlands Bioimaging is part of it. We have created a repository on GitHub. The only added feature is that we would like also to align with the ISA standard, that's investigation study assay. And so we want to basically REMBify ISA or ISAfy REMBI, so that we can take advantage of, for example, the issues that that Matthew was talking about, the fact that, for example, REMBI does not really capture the aspects of a process of and assay and stuff like that. So, yeah. Thank you. I don't.
Awesome, thanks. A lot of you might know I'm replacing Natalie here, and there's a big shoes to fill, but she's been working on a project and I've joined with her for the last couple of months, where we're working with HHMI, CZI, and we're in communication with Kevin and others from BINA, Josh and Jason from OME to try and get this right. We're still in the early stages of it, but the idea is there's tons of great data stored everywhere, but how can you search across all of it at once? There's a long-term vision where if we have this philosophy, if we can scrape all the metadata into a lightweight centralized metadata repository, the pixel data can be stored wherever. It doesn't even need to be accessible. You could still know about data that's stored in some professor's drawer, for example. And with BFF, we've seen that we have already an opportunity to test this concept with the capability of just being able to concatenate multiple metadata files into one system. So we're working with these organizations to just prototype the first kind of internet of bioimage data, just to see how bad things are. If anything, we might run a contest once we scrape some of this data together. Maybe one of the contests would be on metadata harmonization across all the different columns that intend to mean the same thing, but are spread around. But then downstream of that, if you're combining something like zebrafish data with human cell data with EM data, can people do some really new cool science with that? So come to the poster tomorrow, 18th. Thanks.
Element.
Okay, so I just want to talk about collections, viewing ZAR collections, and in particular, viewing thumbnails, because as has been discussed earlier, viewing collections of ZAR images is much nicer if you've got some thumbnails to look at as well. So, and here's a bunch of examples, but in all of these cases, the thumbnails have been pre-generated by some other application, some other infrastructure. But in the case we had talked about this last year of the 2024 challenge, what we wanted to do here was you generate thumbnails on the fly. So you've just got the URL and they're generated on the fly in the browser. So what I did is took the code for the generation of thumbnails used here and put it in a separate library so that other people can use it. Is omezar.js, and the idea here is that you can just take a one-liner, you can go from the URL to an image source, and you can just set that source on the image, and you've got your thumbnail. So having got this library, I've gone on to use it in a couple of the other projects we work on. One is to add thumbnails to the NGFF validator, so that when you view these guys now, you'll see a thumbnail generated on the fly. Another place I'm using this is in AmeriFigure. So this is a work in progress, but the idea is AmeriFigure works as a standalone application without Amero, using ZAR to browse the images, and this is an example of that. All these images are ZAR images. And if you want to work for this standalone figure, but you have just a collection of images, whole load of ZAR images on your laptop, and you want to serve those to use in Figure or in fact any other web-based viewer, the OME ZAR library now supports OME ZAR Finder command, where you just point it at this data. It will traverse all those files, find all the ZARs, put them in a CSV, serve the CSV, and launch BFF all in one go. So then you can browse your local ZARs with thumbnails and copy those URLs to use in any web-based viewer. Okay.